Hello friends, welcome to my YouTube channel, The Microbiology Digest. We are discussing about the viruses and we have come to know that viruses are infectious agents associated with some disease conditions in man and animals. But today's discussion will be talking about the interactions between the virus and the cell or cells of the body when a virus infects a cell in a host. What are the different type of interactions that may take place between the virus and the infected cell? So to start with today's discussion, again I welcome you all to today's discussion of virus cell interaction. I am Dr. DP Bora. So we'll be discussing about the different type of interactions that may take place between a virus and a host cell. Now let us understand what is virus infection. So as we all know, like other infectious agents, viruses they can also cause infectious diseases in man and animals. But viruses, they are obligate intracellular parasites. They always need a living cell to grow and then only they will be able to multiply. So once a virus infects a cell, it has to go through some processes. So already this entire process we have discussed in our replication discussion on virus replication. So the viral infection, it starts with the infection of individual cells, and after establishment of the infection, the virus gets spread through different systems or body fluids to the different sites, and they try to establish their infection. So at when it infects a cell, when a virus infects a cell, first step in the virus infection is the attachment. Means virus has to establish the attachment with the cell. So the replication cycle we have discussed, there are 7 different steps in the viral replication. So infection of individual cell, when we say the infection has already established, only when virus successfully multiplies inside the cell. So this includes the all viral replication steps, namely the first it has to attach to the cell through receptors, then it has to penetrate, then there will be uncoating of the nucleic acid, then transcription, translation, then replication of the nucleic acid, finally the assembly and release of the virus. So these steps will, once the virus completes these steps, then we can say that successful virus infection has established. Now, what is virus cell interaction? What we understand? So virus cell interaction, it is the process where virus as obligate intracellular parasite, as we all know that, it attaches and it entered to the cell and it manipulates the host system so that virus can utilize the entire host cell machinery to replicate its own genome. So the entire events or entire processes involved in this process, it includes the discussion of virus cell interaction. Now before discussing about the virus cell interaction, we'll have to understand some terms or terminologies related to this discussion. First is the cytocidal infection. So what is cytocidal infection? These type of infections, they are characterized by cell lysis. There will be lysis of the cell and there will be production of cytopathic effects or CPE. Then next terminology is the non-cytocidal infection. So here in this case, there will be no lysis of the cell. Non-cytocidal means there will be no lysis of the cell. Then third terminology is the non-productive infection. It is also otherwise called as abortive infection. So this type of infections, they do not lead to production and release of new virions. So normally when a virus establishes an infection, so finally it will be successful in production as well as release of mature virus particles. But if it is non-productive or abortive infection, then there will be no production of new virions. Then next terminology is the permissive cells. Permissive means these cells which will support complete replication of a particular virus. So these type of cells are called as permissive. And similarly, the non-permissive cells, they do not support viral replication and they can block the viral replication cycle at any point. Suppose it may not allow the virus to attach to the cell, or it may not allow, or it may block the replication strategy employed by the virus. So at any point of the replication cycle, the cell may block the virus replication, and this type of cells are called as non-permissive cells. They do not support the viral replication, and thereby there will be no release of the mature virion. Then next is the cytopathic effects. The cytopathic effects means these are the changes, structural changes in a host cell which has been infected by the virus. So these are the structural changes in the host cell, which is due to virus infection. Once a virus successfully establishes an infection in a cell, along with the virus multiplication, cells will show some of the changes in its structure. So these changes, which are due to virus multiplication or replication, these changes are collectively called as cytopathic effects. So in short, it is called as CPE. Next is the DI particles. DI particles already we have discussed. This represents the defective interfering particles. So these are actually deletion mutant of a particular virus, and they generally have a defect in the viral genome, and thereby they cannot replicate by. So these are defective interfering particles or DI particles. Now coming to the actual topic, type of virus cell interaction. So when a virus infects a particular cell, there may be three types of interactions which may take place between virus and the cell. First type of virus cell interaction is the cytocidal type of interaction. So cytocidal type of interaction, already we have discussed what is cytocidal infection. So here there will be due to replication and multiplication of the virus. So cells will undergo lysis and there will be cell death, and there will be production of virus specific cytopathic effect. Okay, this type of infections or interactions are called the cytocidal interaction. In cytocidal interaction, invariably there will be production of infectious virion, because once the viral replication cycle is complete, there will be production of new virions and these virions will be released from the cells through lysis of the infected cell. So there are many viruses which undergo this type of interaction. For example, alpha herpes virus, enterovirus, reovirus. So these viruses, they shows cytocidal type of interaction with the cell. So second type of virus cell interaction is the non-cytocidal type of interaction. So non-cytocidal type of interaction means here there will be no lysis of the cell. So again, non-cytocidal type of interaction may be of two types. First type of interaction is the persistent type of interaction. Persistent type of interaction, two type of persistent infections may be there, interactions may be there. One is persistent productive and another one is persistent non-productive. So in persistent productive, there will be no production of cytopathic effect, no or little, very little metabolic disturbance will be there. The infected cells, they will continue to divide, but there may be loss of some special functions of some cells. So these are the characteristics shown by persistent productive type of infection. So as it is a productive type of infection, there will be invariably production of infectious virions in this type of interaction. Means persistent productive type of interaction leads to production of infectious virions. So examples are there: pastiviruses, arenaviruses, rabies viruses. These viruses, they shows persistent productive type of interaction. Then second type of persistent infection interactions are persistent non-productive. Means here there will be no changes will be there will be no production of infectious virions, and one very good example is canine distemper virus. So this virus, it shows persistent non-productive type of interaction. Then second type of non-cytocidal interactions are latent interactions or latent infections. So here, what is the effect on the cell? So in latent infections, the virus infections will be present in the cells, but it will not exhibit any sense. So that is why the literal meaning of latent means it exists but not exhibited. Means the virus is there, but it is not able to show any changes, or there will be no production of any infectious virus particle. So generally, this type of infections are or interactions are found in case of herpes viral infection. The third type of virus cell interaction is the transformation. So in transformation type of interaction, there will be alteration in the cell morphology. The cell morphology will be altered, and the infected cells, they can be maintained for indefinite times, and they may produce some type of tumors when these cells are transplanted to some experimental animals. So in mostly these viruses, the oncogenic viruses, they shows this type of interaction, that is transformation type of interaction. So both DNA and RNA viruses, they can be oncogenic. So for example, among DNA virus, polyoma virus is the example, and among RNA virus, the sarcoma virus, this is the example. So these are different types of virus cell interactions which may take place when a virus infects a particular cell. So what are these type of interactions? First one is cytocidal, second one is non-cytocidal, third one is transformation. So non-cytocidal, there may be of again two types. One is persistent and second one is latent. So persistent may again be of two types: persistent productive and persistent non-productive. Okay. So these are the different types of virus cell interaction. Now, cytocidal changes in the virus infected cell means these changes which are due to virus multiplication, they are collectively known as cytopathic effects. Means these are virus induced alterations in the cell, or these are the changes brought about by virus in the cells. So these cytocidal changes, what may be the changes we may see when a cytocidal in type of interaction may takes place between virus and the cell? First is the it kills the cell, so invariably there will be death, cell death. Then there will be production of cytopathic effects, and these cytopathic effects, they are characteristics for a particular virus which is utilized for recognizing the propagation of the virus in the laboratory. So we can say whether a particular virus is multiplying in a particular cell or not by observing the virus specific cytopathic effects. So again, these cytopathic effects may be seen in different ways. So some of the examples, that there may be ballooning of the cells, there may be rounding of the cells, there may be clumping, there may be cell lysis, there may be aggregation, there may be syncytia formation, or sometimes there may be formation of inclusion bodies. So these are the different types of cytopathic effects which may be seen when a virus produces cytopathic effects when it multiplies in a cell. Now, how these viruses, when they multiply inside the cell, how they can damage a particular cell? What are the mechanisms by which virus can damage the cells? So first mechanism is the virus may inhibit the host cell nucleic acid synthesis. So there may be inhibition of the host cell nucleic acid synthesis. Then there may be inhibition of host cell RNA transcription. There may be inhibition of host cell mRNA processing. There may be inhibition of host cell protein synthesis. There may be production of cytopathic effects. There may be production of structural changes involving the cell membranes. There may be fusion of the membranes due to formation of syncytium, and there may be heme agglutination or heme adsorption. So these are the different mechanisms by which a virus can cause damage to a cell, infected cell. Now we have come across one term, that is inclusion bodies. So inclusion bodies, these are virus specific intracellular mass which are produced during the course of the virus replication. So these are intracellular mass, and they are produced during the replication or multiplication of the virus. These inclusion bodies, they can be observed under light microscopy, and many a times they serve as an important diagnostic tool for some of the diseases. So these inclusion bodies may be diverse. There may be accumulation of viral components. There may be crystalline aggregates of the virus, or inclusion bodies may be a result of degenerative cellular changes due to virus infection. So any type of inclusion bodies may be formed by any of these three ways. The viral components may aggregate, or they may aggregate to form a crystalline structure, and they may be result of degenerative changes due to virus infection in the infected cell. So by any means or any of these three ways, inclusion bodies may be formed. These inclusion bodies may be intranuclear, they may be formed in intracytoplasmic. So two types of, based on the location, it may be either intracytoplasmic or it may be intranuclear, or again it may be either acidophilic or basophilic. So these are some basics about the inclusion bodies. Now another term called apoptosis. Apoptosis, already we know that apoptosis means this is a programmed cell death. So there are some viruses which can induce apoptosis. So it is programmed cell death. Already we have studied in our immunology discussions. So it is one of the very important mechanism of host defense system. It is a part of our immune system, and some viruses, they, as I said, they may have the capacity to induce apoptosis. How? They may act directly on the viral proteins, or indirectly they may act on any of the cellular processes. Okay, so examples are there. There are many viruses, for example adenovirus, charcovirus. These viruses, they can induce a programmed cell death, which is called as apoptosis. So here it is important to understand the difference between apoptosis and necrosis. Necrosis is another process where there is also cell death. There is difference between apoptosis and necrosis. So first difference is the apoptosis is a physiological process and necrosis is a pathological process. So likewise, there are different points where by which we can differentiate between apoptosis and necrosis. Next is the interferons. Interferons, we all know that these are the glycoproteins. They are heterologous group of glycoproteins. These proteins are produced as a result of viral infection. So these are a group of heterologous proteins produced by virus infected cells in vivo. These interferons, or this group of proteins, were discovered by two scientists, Isaacs and Lindenman. So they discovered this group of proteins in the year 1957. These interferons, they may protect uninfected cells against the same, or sometimes it may protect the cells from unrelated virus. Also, basically there are three different classes of interferons: interferon alpha, interferon beta, and interferon gamma. So we can differentiate very well among these three. So if we see some of the properties like principal source, inducing agent, glycosylation, functional form, principal activity, then mechanism of action. Details about interferon we have already discussed in our immunology class, and you can please go through to know details about the interferon. So at last, there are some general properties of interferons which we should very well be aware of these properties, and I have included here some of the properties of interferons. Please go through this briefly. These are, as I said, interferons are a family of glycoproteins. They are resistant to heat, they are stable, they do not have any direct action on viruses, and most importantly, their activities are not virus specific. Okay, interferons induced by one virus is effective against many other viruses. They are not virus specific, rather they are specific to the host. So similarly, there are some other properties which we should understand, or which we should know to understand the interferons in a better way. So this is what I wanted to discuss or share with you all today, and I hope this information will be helpful. And if you find these are helpful, please subscribe to my channel and give your comments so that more and more topics can be included for discussion